this video, we will be covering genetic circuits, a fundamental concept required to understand how to reassemble cells so that their functions can change. Genetic circuits consist of four key components: the promoter, ribosome binding site, coding region, and terminator. Promoters are the first component of this assembly line, as promoters are able to provide the RNA polymerase with the location of the gene targeted for transcription. In other words, the promoters are able to send the RNA polymerase to the location of the DNA sequence that will be genetically engineered. There are two main types of promoters: constitutive promoters and controllable promoters. Constitutive promoters are able to continuously code for the production of a protein with no regulation. Controllable promoters are promoters that can be switched on or off by a specific agent. The second component is the ribosome binding site, or RBS. RBS is a sequence where the ribosomes attach to the mRNA to conduct translation. In other words, a ribosome attaches to the mRNA at the ribosome binding site sequence and translates the mRNA into a series of amino acids. Without the RBS sequence, the ribosomes wouldn't be able to position themselves properly onto the mRNA and therefore fail to translate anything. Once the ribosomes attach to the mRNA to conduct translation, the coding will begin at the gene of interest. The final component is a terminator. A terminator sequence terminates transcription. The particular sequence of nucleotides in a terminator causes the DNA at the end of the gene to fold, which blocks further progress. To summarize, genetic circuits, which are composed of promoters, RBS, gene of interest, and terminators, are utilized to genetically engineer organisms.